For a long time we've noticed that HER2 positive breast cancers have high levels of immune infiltrate. This immune infiltrate was prognostic, so this led us to hypothesize that changing the immune microenvironment can further improve survival. So when checkpoint inhibition became available, we asked if the company would evaluate the combination of pembrolizumab and trastuzumab. So this is a study, it's a single-arm study. We looked at pembrolizumab and trastuzumab in trastuzumab-resistant advanced HER2-positive breast cancer patients with the hypothesis that pembrolizumab may reverse some trastuzumab resistance. It was a single-arm study. There were two cohorts, PD-L1 positive cohort and a PD-L1 negative cohort. And all patients were centrally screened for PD-L1 status by the Merck test, and we did central screening of HER2 status, as well as looked for levels of immune infiltration. And we found that the study met its primary endpoint. The primary endpoint was objective response rate, and we achieved the required amount of pre-specified responders. And the nice thing was that the patients that responded had durable disease control. So they were nearly 1 year, median duration of 1 year on treatment, so without any chemotherapy. So we thought that was really a good result for a proof of concept study. So I think the stronger biomarker is probably the level of immune infiltrate. And now we know that this varies by site of your disease. So lung disease has more immune infiltrate, breast has more, patients with liver disease have less levels of infiltrate, suggesting that they have a less chance of responding. So I think that both components actually help us piece the information together. So if you have a patient with high level of TIL or immune infiltrate and no PD-L1, you're probably going to question that result a bit. So I think both pieces of information help. So I think in HER2 disease we know what the driver is, so I think we have to have more effective targeting of HER2. So this may be with more potent combinations or some of the newer drugs that are being evaluated in this setting post-pertuzumab. But obviously I think we found that as little as 5% could enrich for responders. So I suspect the earlier we go with regards to earlier lines of treatment, more patients are going to have higher level of TIL infiltrate. So I think certainly the earlier we go, the higher chance we'll find patients that are going to achieve durable responses to pembrolizumab. And then if we add a more potent anti-HER2 agent, we might be able to get away without any chemotherapy. That remains still to be tested.